3, 2, 1, and here we go. Hi everybody, welcome to Genomics. So this week we're going to see evolutionary genomics and a little bit of population genetics, population genomics, for the purpose of introducing the next class, which will be on GWAS. So this week we'll kind of prepare the ground for GWAS, and we'll see different topics. But before we do that, I wanted to touch upon a couple of topics that are unrelated with today's class. So the first is an announcement that I was asked to pass on to you, and that's for note-taking services. So as we go back in person, people need to take more notes in classes, and for some people who have disabilities, they have the right to have someone who's helping them take notes, or maybe who's taking notes for them. I'm not sure exactly how that works. And these are volunteers, so these are other students who get to take notes for someone else. And so they have asked me to announce and to advertise for you a new career opportunity as a note taker. So that's volunteering. Actually, that's not really a career. But if you're interested, if you want to help out people who have disabilities and who require a note taker, then you can just go to the website. So here, that's the main website, Peer Note Taking Services, and you've got this kind of advertisement that they have at this link. So if you're interested, or if you're just curious, please go ahead, have a look. That could be something useful. So that was the first announcement. Now, more directly related to the class and to the content of what we're doing, I'd like to give you some background information on the blog post. So from now, almost everything we do will be for the blog post, for the final exam that you have in this class. There's only one relatively big roadblock. It's the whodunit. As you've seen, I've published it, so I apologize for having put the questions of the previous year. There was some mess up that really happened for the midterm exam, is that the content of the previous year leaked into the content of this year in a non-optimal way. So I have corrected the whodunit. Now the questions are the ones that you have to answer. And in principle, you have access to the data. Each of you has a different data set. So even if the question is the same, bear in mind that the answers are not. Okay, so don't go copying the answers of your friends, because they're not necessarily applicable to your data set. Even though the questions are the same, I don't expect that the answers will be the same. So you have access to the data, and you have to access it the same way as you access the data for the midterm, which means on the cluster. But you do not have to use the cluster. I mean, there's nothing to do on the cluster at all. It's just because it's easier for me to dispatch 40 exams this way, rather than having to send you individually by email the data that applies to your questions. If you can't access your exam in the next days, just tell me, and on the case-by-case basis, I can always send you the individual sheets. It's just that doing it 40 times actually takes too much. So the whodunit, I've postponed it by 1 week because, of course, I've maybe kind of created some situation for some people if they have already started to do the whodunit, but also because I realized that you guys have a lot going right now, so it was a bit better to postpone it by 1 week, which will be then after the last class. Right? So you finish the last class, you still have 2 days or something like that to answer the whodunit. And then that will maximize the time you have for working on the blog post, because after the whodunit, there's nothing else to do than just writing your blog post and reading other people's blog posts. So except the whodunit, then most of our work will be about the blog post from now on. And I wanted to give you a little bit of some background, the way things are going to run, but also some tips and tricks on how to read a paper. The first time I read a paper, I was something like 20, and it was an easy review on photosynthesis, and it took me approximately 2 weeks to just go from the title till the end, and I still had understood nothing at all after 2 weeks. So I remember the first time you read a paper, it feels like, oh my, that's complicated, that's hard, and I don't know how people can read more than 2 per year or something like that. Nowadays, it takes me approximately 10 minutes to read a paper, and so that just shows that between the very first time you read and when you're an experienced researcher, a lot happens. Like, you can develop ways and tricks and methods to read fast, to know what you're looking for, and to understand faster the content. So that's what I'd like to do with you, is to share a little bit what I've learned between my first paper and what I do now when I read a paper. But first, let's talk about a little bit what you have to do for the final exam. My recommendation is that you should read the demo post. It's linked in the main page of the final exam. So the final exam is already in principle available on Quarkus. I'll double check that the link is available. So I've written a demo post last year that corresponds to something that's, let's say, average. Okay, so this is not the best blog post ever written by far. I didn't want also, to do everything for you, but that shows the basic structures that you have to follow. And so this is the article that's called "On the Wings of the Mayfly: 350 million years in one transcriptome." That's my blog post. But better than that, you can read the blog post from the other students because they're online. They're much better templates than the one that I provide. And so when you read the blog post, the demo blog post that I wrote, you'll see a couple of things. The first, and that's the most relevant for you right now, is that the TLDR really has nothing to do with the abstract of the paper. All of you have an abstract in the paper, and that should really, really not be used to make a good TLDR. My recommendation is don't take the abstract, don't shorten it to make a TLDR. That's a very poor strategy. That's really not going to make a good TLDR. The purpose of a TLDR is really to catch attention and to kind of reveal something without revealing too much. You really want to be enticing. The purpose of an abstract is not that at all. It's to summarize the paper so that people don't have to read the whole paper. It's actually, the purpose of an abstract is more to tell you, don't read the paper if you're interested in that topic, right? So because we're not talking about it. We're talking about this, this, and this only. So if you're interested in something else, don't go wasting your time on reading the paper. So it's the opposite of enticing an abstract. It's just to say, that's what you'll find in the paper, and if you want to commit to spending a couple of hours, that's all you're going to find, not more than that. The TLDR is the reverse. You want to convince everybody that you're awesome and that you've written the best text in the world. So don't just use the abstract of the paper to write a good TLDR. That's not a good strategy. You have to find other ways to make the reader interested in what you want to say. The second thing you'll see in the demo post is that there are a couple of hyperlinks to other sources. That's really what makes a blog post, is that it's not references, even though there are references, is the ease with which you can just click and then link to the Wikipedia page. Everybody's able to find something on Wikipedia, but it's kind of nice to make it easy for them to just click and then get the content in just one click. Also, that's important to link knowledge that you're going to put to the rest of the world. That's a little bit what internet is for anyway. The third point is that the figures that I chose are not the ones that are in the paper. I either created new figures or I modified figures from the paper to make the point, because the figures in the paper want to make a point that is very nerdy, very specific, and I don't want to write nerdy, I want to write for a wider audience. So I also have to simplify the figure, like removing the part that doesn't make my point, simplifying the axes, simplifying the units, or something like that. It's okay, as long as you say what is what, you don't have to give all the detail. So you'll see on the demo blog post how I modified the figures from the paper to make them more adapted to the audience that I expect for the blog. And you also see, like this sentence, our analysis at first approximation, blah, blah, blah. The sentence that is here is actually from the author, from the first author of the paper that I'm commenting. So how did I get that quote? Well, I wrote to her and say, hi, I'm Guillaume, I'm a teacher, and I would like to use your post, your work, to make a demo blog post. Is that okay with you? She said, yeah, fantastic. She sent me plenty of extra material, just so that I have enough. And she sent me, among others, this sentence that I just pasted in the text. You have to do the same. Well, you don't have to say I'm a teacher, you have to say I'm a student, of course, when you contact the authors. But it's really nice when you can put inside the blog post a comment from the authors themselves. That makes it first a little bit more personal, like no one else than you can put these things, because this is new material. That's more interesting. Second, it's nice to see what the authors themselves have to say about their own work. And third, that connects you a little bit with the scientific community. You got to start now. So this is expected from you. It's a bonus point you can get for that. So if you don't do it, you won't be penalized. But if you can, it's kind of nice. Don't expect everybody to answer your questions, because some authors are just like, they've got big ego, they've got no time, etc. So you can't expect everybody to answer. But sometimes, I'd say a good 50% of the time, when you just be polite and say, I loved your work, it was so wonderful, people say, thank you. And then they'll give you some more. So if you just be polite and nice, I think 50% of the time you can also get a good. So that's for the structure. Now you must be at the stage where you maybe have read the article in full, maybe not yet, probably still struggling with making sense of it. Most of the articles, even if they're short, are hard to understand, and that's normal. That's state-of-the-art knowledge. It's hard to understand. So here are a couple of tips that will help you read more efficiently. The first is what I call, and nobody else calls it this way, the magic paragraph. That's the one that will give you the key to everything else. So what is the magic paragraph? It's the one just before the results. Okay, so that's the paper that I was commenting in the blog, Genomic Adaptation to Aquatic and Aerial Life in Mayflies and the Origin of Insect Wings. So the abstract starts here, etc. And then there is this clear-cut demarcation between the introduction and the results. The last paragraph of the introduction, the one just before the results, is the magic paragraph. Why? There, the authors explain what they did in relatively simple terms. This is usually better than the abstract to understand what they did. The abstract is a little bit for marketing, to say like, we did this, and if you're interested in that kind of research, that's awesome. But it's not really factual. Here, in this paragraph, it's expected that the authors really tell you what they did. We did this experiment in order to find that, because we thought that. Right? In the abstract, they will just jump to conclusions and tell you what the conclusions were. But in the magic paragraph, they already start to explain in relatively concrete cases, concrete words, what they've done, what was the reason, what were they looking for. Because that's what you need to be able to make the content a little bit simpler. So read again the magic paragraph in your article. It can be, sometimes when they really did a lot, it's 2 magic paragraphs, but in general it's just 1. And make sure that you understand that magic paragraph. This is actually where you can start reading the paper. Not even the title, not even the abstract. Read the magic paragraph. This is really what they did and kind of what they expected to find and what they did it for. The second tip, which is kind of not obvious, I'm saying these things because they're not obvious. It's not obvious that the paragraph just before the result is the most important, at least for me. Second thing is the perspective. Where's the perspective? In general, and again that can vary, is the second paragraph of the discussion. And you can have 2 paragraphs of perspective, but so why is it there? So the perspective is usually the second paragraph of the discussion. The first paragraph is just a recap of the results. The authors place the results in context right after the recap. So yeah, think about it. They did all this beautiful work, that's wonderful, that's magic, but it's complicated, so at the discussion they need to regroup and they say, we did this, this and that. So the first paragraph in the discussion is really just saying what they've done. So it's, again, if you want a copy-paste of the magic paragraph almost. To just, okay, we told you we would do it, well, we actually did it. So that's the summary of what they've done, but also what they found here in this paragraph. Now, when you're interested in what that means, they're going to follow up right after that. They first have to say, we found this, what it means is that. So that's usually right after, in the second paragraph. Now, it's not always exactly the second paragraph, but this is more or less where, if you want to know the perspective, like how they interpret the results within the grand scheme of knowledge, you're going to find it here. Okay, you're not going to find it at the end of the discussion, not at the very beginning, but right after they have summarized what they found. Okay, they have to summarize what they found first, just to regroup, and also because not everybody reads the results. So, like for people who've teleported to the discussion, they need to say, we found this and that. So, please bear with us. Now we're going to explain to you why this matters. Why this matters is usually here. It's not at the end of the discussion. So another paragraph that I advise you read and reread until you get it. When you get it, you understand a little bit how they understand their own results. So figure 1 is usually the most important because it's a graphical recap of the study before they did it. Okay, it comes first, not last, it comes first. It always contains the model organism or study design or technology, etc. So they want to make sure that the reader has enough information to understand what they're going to do. So they kind of find a graphical way to express that. So in my case, it's this. So they show you a little bit what the organism looks like, why this is interesting. So it's a mayfly. They've got interesting life cycle that has, an aquatic form and also a flying form. So they will tell you everything that is interesting to know about their system. Also, they will tell you what their technologies are at that point, what really matters for the main studies for the main experiments that are in the study. It's all in figure 1. They cannot really bring that later than figure 1. Right? Like they have to tell you that's what we're going to use, and then they use it. So you have a nice recap of the method, the tool, etc. in figure 1. So make sure you understand figure 1. If you don't understand figure 2, 3, 4, it's not that bad. Figure 1 is really the figure that explains the methods. The legend is usually technical, but it really helps understand the figure. Right, just make sure you understand your figure 1. Tip number 4, don't read the methods. Just don't. You probably have tried, you've seen the headache you get, right? And it's just a headache. It's never going to help you, ever. Why? Because it's not for you. This thing is not written for you. You have to understand that a scientific paper is not written for one reader. It's written for multiple readers. And each reader has got their section that's for them. So the title is for the journalist, the abstract for the scientist, etc. The text itself is for scientists, young scientists, students. But the methods, they're not for you. They're for people who want to reproduce their results. They're for people who actually, even for their own lab, so that people remember what they did. So this thing is only useful for people who plan to either challenge the results or reproduce them, because these methods, they're supposed to give enough information so that anybody can reproduce exactly what they did without having to contact them, because people hate to be contacted about details, so that they can answer. The details are in the methods. So don't read them, they're not for you, okay? They would be for you if really you become an expert enough that you can think, oh, but wait a minute, how many millimolar of KCl have they put in this experiment? Now this is really important for my blog post. I don't think he's gonna get there. Nobody really cares about these kind of questions anyway. So don't read it, skip it. There's no information that's interesting for you here. Don't waste your time. So that's it. These are 4 relatively simple reading tips. They're not absolutely essential, but it's just that no one will tell you that. So here they are. Another thing that's quite important and interesting for you to get working on that is that our librarian, Sarah, has assembled a useful guide. So that's the link here. To write a blog post. She gave the information about the inverted pyramid that Kyle showed last time. So that's a very standard trick. It's not a trick, actually. It's really the basis of how you have to write information in general. You have to follow this inverted pyramid structure. So she explains a little bit what is good style. I 100% agree with her, with whatever she wrote there. So if you read that and you understand it, it's going to be a good framework to get started with your blog post. So you can read the guide here. It's also available at library resources on the Quercus page. You have multiple links to this guide. So read it. It's kind of short, but she's following her own advice the way she wrote it, so it's easy to read. And one last thing about the blog post before we go to the class proper. You can publish it, you cannot publish it. This is really up to you, and I will not ask before grading. Okay, so first, I don't want to know whether you're going to publish it or not. I don't want it to influence the grading at all. So you keep it for yourself whether you want to publish it or not, and I will not ask that question until I have posted all the grades and they're final. Okay, so that will really not at all interfere with your grades. If you want to publish online, that's good. If you don't want to publish online, that's also good. This has nothing to do with your grading, so I want to make it clear. So why would you publish something then if it's not getting you any bonus points? Well, it's up to you. But I wanted to share this screenshot with you. That's one of the blog posts that was published last year, and so it's one of the comments that was there from Michael Romanov. Congratulations, Brantha, you did a great job. As one of the key co-authors of this study, I would love to have you on board of our research team. So Michael happens to be one of the authors of the article that she wrote about. And you can see that sometimes when you take risk, good things happen, right? So I'm not sure that Brantha was interested in working with Michael, but she got an offer for free for a work that was actually at the undergrad level to say that you are already invited for a job interview or maybe more to work in the lab. That's actually a very good head start if that can happen to you. So if you're still on the fence whether you should publish or not publish your work, think about it. Like, there can be good things happening to you if you publish your work. If you don't publish your work, no special good things will happen to you. So remember that you cannot win the lottery if you don't play. That's basically the one reason to publish is, well, you don't really have much to lose, really. I will ask the volunteers after the class is over. You can wait to see if you're satisfied with the final version of your work, and there's nothing to decide now. Okay, so if you really like your work at the end and you're ready to share it with the world, just go for it, publish it. If you don't like it and you think, yeah, but just like not happy with what I did, well, don't force it either. You know, like you're not proud of yourself, don't share it. You will also be allowed, even encouraged to further modify your blog post after the final grade if you want to publish it. You have as much time as you want to improve it, so that it's kind of publication ready. But these are things I would like you to think about, but don't tell me if you want to publish them or not. I don't want this to interfere with the grading at all. That's all I wanted to say about the blog post for now. That should really give you a bit of perspective, especially for writing your title and TLDR. You will be able to change them, titles and TLDR, for your final version of the blog post. I'm not going to evaluate them again though. So just for the consistency of the whole blog post, you will be able to change the final title and the final TLDR in the final version, but I won't grade them again. Are there any questions about this? Online, are there any questions? Doesn't look like it. Okay. So if everything's clear, let's get started with the class proper, evolutionary genomics. So why study evolution, especially in the class in genomics? Well, one thing is that the curious aspect of the theory of evolution is that everybody thinks he understands it. This is a famous quote by Nobel Prize winner Jacques Monod. And so one reason to study, the reason to study evolution is because we don't really understand it. Okay, we think we do. Everybody thinks they understand evolution, but in reality, we don't. Also, evolution is important because it tells us what is conserved, and what is conserved is usually conserved because it's important. Okay, so here you can remember what we've seen in the first class. This is a screenshot of the UCSC genome browser, and you've got this conservation score, this FastCon score here in general, and you can see that the conservation is high for exons and low for introns because exons matter a lot. They code proteins. Introns matter much less. They're removed, so it doesn't matter so much what is there. But you can see that sometimes you've got conservation within introns. So why is it so conserved if it's anyway removed and doesn't even contribute to the protein? Just the fact that something is conserved when we don't expect it to be conserved is an important information, and that means, oh, now that becomes interesting. What's happening here? It's worth having a look. Maybe there's something to discover there. So conservation by itself is really a very important criterion for studying something. If biology wants to keep it this way, it's maybe because there's a reason. So evolution is kind of a guide for us to know what we should study, what we should not. You know, like something that's totally not conserved, so it's just random filler. Like there needs to be some nucleotides, and happens to be a G, a C, a T, and an A here, but it could be anything else. Because when you replace it by something else, nothing changes. It doesn't matter. You know, the biology is the same. Also, another reason to study evolution is that dinosaurs are really cool. Of course, everybody knows that. So you're probably familiar with Darwinian evolution, and my advice is, if you haven't done so already, read The Evolution of Species. It's actually a good book. It's not boring at all. I made a mistake in my career. I made many mistakes in my career. But one of the biggest is that I read it too late. I was already older than 30, because I thought it's going to be written like Shakespeare, you know, like in verse and rhymes and all that stuff. Not at all. It's actually very fresh, and you'll see that Darwin was really a brilliant person, and the way he thinks, the way he describes his thinking, I found very useful for myself to think about this stuff. I also realized he's much smarter than me. Maybe I should listen to what he has to say. So that's one version of evolution, and that's the one that everybody hears about. The one that is less popular but equally important is called neutral evolution. So the neutral theory proposed by Moto Kimura in the 1960s posits that most genetic variants do not evolve through the Darwinian paradigm of natural selection. Aha, that's interesting. This theory is often misunderstood. It merely says that there is no fitness advantage to either allele. It does not say that the changes are random in the sense of unbiased or uniform randomness. Okay, so the difference between Darwinian selection and neutral theory is not that the neutral theory says that every change is completely random and doesn't matter. It just says that these changes in the sequence do not change the fitness of the organism. Okay, that's really what neutral evolution is. The changes, they can't be biased. They can be anything. The only thing that they cannot do is change the fitness of the organism or the population, if you will. So if something changes the fitness, then it will be subject to Darwinian selection, just because the fittest will survive. Duh. If something does not change the fitness, then it cannot be subject to Darwinian selection, because there's nothing to select for. Everybody's got the same fitness, no matter what the variations are. So think about it. The SNPs in the human genome, right? Are they mostly evolving according to Darwinian selection or to neutral evolution? Go ahead. Neutral, right? We've seen that most of them, you can have any of the two variants. It doesn't change your fitness at all. You're not sick or infertile or anything, depending on the variant you have. It's only some variants that have an associated fitness cost that make people sick. It's a small minority of SNPs. Okay, most of them are actually neutral. So based on these ideas, people started to have models for how things can evolve, and we can use that. So one particular model of interest is the model of molecular clocks. So the divergence between sequences is proportional to the time they diverge. Okay, like you have two species that split, like a speciation event. Remember what we saw with the ribosomes, for example, the ribosomal RNA. So these two sequences will start to have their own life, because the two species now live in different contexts. They have their own life, basically. So the two sequences will start to diverge a little bit, or maybe a lot. And the older was the split between these two species, the more time there was for evolution to change the sequence. So you can expect that when two sequences are very different, then probably they split a long time ago. And when they're not very different, then they split not that long ago. So some proteins evolve fast, some evolve slow, but this is often a constant. Okay, so like the ribosomes, the ribosomal RNA evolve extremely slow because they need to be extremely well conserved. And some others evolve extremely fast. Some sequences evolve crazy fast. But what's really interesting is that it's either fast or slow, but that's constant most of the time. So here, this is the first paper that discovered molecular clock phenomenon. So they're showing here, for example, the cytochrome C, and they know how long the species have been separated, when the split happened, and so they can measure here on the x-axis the number of million years of separation, of individual evolution of the proteins, and on the y-axis they can measure how many amino acids are different between the sequences. And you can see that for cytochrome C, they accumulate mutations, changes in the protein sequence, relatively slowly versus actual time. And for other protein, like hemoglobin here, or what is this one, the fibrinopeptides, the evolution is faster. But still, every time, the point is on a line. Okay, like these are experimental points that they see here, plus the number of differences that they have observed experimentally on the y-axis. And on the x-axis is how long the proteins have been diverging separate from each other in terms of millions of years. Okay, some proteins evolve slow, some proteins evolve fast, but they evolve at a constant rate. They don't switch category, going from fast first and then slow. And if they evolve fast, they always evolve fast, and if they evolve slow, they always evolve slow. So this property is not always true, though. Example, humans evolve slower than other great apes. So like the apes population has diverged, like there's the gorilla, chimpanzee, orangutan, and humans. And so you'd expect that everything evolves at the same time, at the same speed, but humans evolve slower. But it is often used to measure how much time passed since sequence diverge. Okay, like these differences in speed are not crazy huge. We evolve a bit slower than other monkeys, but let's say the difference is not that big a deal. You can still make approximate, you can measure time relatively approximately with this molecular clock hypothesis. So how does that work in practice? It looks a little bit like that. You can sometimes see these phylogenetic trees that are here, and instead of being like completely symmetrical with no information, the length of the branches matters. Like you'd say that one branch is longer than another, something like this fish here. You can see that the branches are a certain length here. So how do you make the evolutionary trees have a certain length for their branches? The time estimates that you sometimes see on the branches of a tree are usually based on the molecular clock hypothesis. That is to say that the protein evolves at the same speed on all the branches. Okay, you use actually one particular protein or one particular sequence, like the ribosomal RNA, like we've seen, and then you make the tree based on that sequence. And the length of a branch will just be the number of differences between the common ancestor and the species that is here. So that the total path, if you want to go from, for example, tuna to human, you would accumulate all the differences here on that branch, plus accumulate all the differences here on these branches, and that will give you the total number of differences between the sequence in the tuna and the sequence in humans. Okay, like the length of the branches is really proportional to the number of differences of the sequences, and we assume that it's also proportional to the time of divergence, so that we can say the time when human and tuna had a common ancestor would be that much based on the evolution of that particular protein. In this case, the length of a branch is proportional to the number of differences between the sequences or between the ancestors. So here's a stop and think. How does the generation time affect the speed of evolution? Okay, like if suddenly an organism starts to reproduce earlier or later, is that going to have an effect on the molecular clock hypothesis? Would it not? Think about it. I'm not giving the answers, just for you to think. Right, is that clear about molecular clocks? Or you want me to go over some aspect once again? Seems to be clear. All right. So we've got now this case where we say, okay, most of the time the SNPs in the humans, they're neutral. Selection still exists. It's still a very, very strong phenomenon. We cannot just discard it because it's not rare at all either. So how do you differentiate between selection, like evolution by natural selection, or evolution by just neutrality with a Kimura hypothesis? Well, the sad answer is you can never be totally sure. Right? These things happened a long time ago. We cannot really measure fitness easily in organisms that are dead millions of years ago. So we can make a hypothesis and we can find consistent models, but we can never answer for sure. And one thing that one of the main ideas for the evidence that something is under selection versus neutral evolution is speed. Allele frequencies change. The question is how fast that happens. If an allele takes over the population very fast, it is likely to be due to selection. Okay, like the guys who have the wrong allele die faster, so the other ones will take over the population really fast. If it's neutral, it's going to be, the allele is going to be increasing one direction, one generation, decrease the other one, but it's random. It's more a random walk, so it's going to take time before accidentally it takes over the population, but it never intended to. Okay, it didn't try to get there in any way. So if this happens much slower, then the changes are probably random. Okay, so like one of the main ideas is if an allele takes over the population really fast, then this is intuitively the idea that probably was based on selection. If it takes the population in billions of years, you say, well, selection sure took its time, and that's not really consistent with a model of selection. Okay, so here is a little sketch where you have chromosome here, which is depicted in black, and the allele with two variants here that could be either light blue or dark blue. And if you make a sampling of different sequences initially, and you see that this light blue allele is rare, and then you wait a little bit, like a couple generations later in the same population, you see, oh my God, this is now much less rare. What has happened? Like, is it really just because there is a selective advantage in having that particular allele, and then it takes over the population, or is it just expected by random chance? And the question is, what is expected by random chance, and what is fast? What is faster than expected? How does one decide that? Well, there are many ways, and one way that kind of is important to understand, at least conceptually, because it will impact on things that you'll see next week with GWAS and other concepts, is recombination. Like, you can use recombination to say what is rare versus what is not. So you have this concept of selective sweep. It's when one particular allele takes over the population because it's selected for. That's called a selective sweep. So how can you detect a selective sweep? Well, it's that if the allele that's under selection, the pale blue one that's here, is going to take over the population, it means that every organism that does not have this allele is going to die or not reproduce, or slower, right? Then this pale allele is going to go up in proportion in the population. But the alleles next to it are also going to go up. Okay, it's like the whole thing is going to go up. In reality, what happens is that everything else goes down. That's selection. Usually, that's killing the individuals that don't have the right stuff. And just because that allele was initially associated with the two other pale alleles here, if you see something like that, okay, like at the second time when you measure the increase in that allele, then it's always associated with the same alleles as it was initially. Well, that looks like a selective sweep because there was not a lot of time for recombination to create new genotypes. Okay, whatever, like the time it took for this allele to go from just one to almost everything was so fast that they did not allow recombination to create new genotypes. Like, it's the whole genotype that's here that's present all the time here. Okay, that's scenario number one. Scenario number two, this blue allele takes over. It's the same proportion as here. Okay, but this time you can see that you got a little bit of everything. You got a little bit of a light orange and light green together with the light blue, but you also have the dark orange, light green, and also dark orange, dark green. You got a bit of everything. So obviously recombination had enough time to break the associations here and to create these new, this kind of new genotypes that are expected when the population is at equilibrium. So when the selective sweep happens really fast, there won't be time for recombination to really break the initial associations. But if this takes time, takes forever, then recombination will happen all the time in the population, and you're going to maintain the equilibrium at the other loci here. Okay, together with the fact that this one is going up, that was maybe random because the other ones that are just nearby haven't really gone up. It didn't take. It went slow, basically. That's the idea. So recombination is a way to measure time. In scenario number one, the two flanking alleles also take over the population. Okay, meaning that there was little time for recombination to act. The pale blue allele is probably under positive selection. In scenario two, the flanking alleles do not take over, so the ascension of the pale blue allele was slower. It is probably not in the selection. That's not a rock solid argument, but that's an idea that one way we can think of selection is that it will push the allele faster than expected in a model where it's just random. And one way of comparing that is with other biological mechanisms. We cannot compare the speed of evolution of an allele in bacteria versus humans, which is like very different orders of magnitude. But within an organism, you can compare the speed of evolution of an allele with the speed of recombination. They're on par. They're in the same order of magnitude at least. So recombination is a very important concept. Yes?
I just have a question with the selective sweep. How do we know that the flanking alleles are associated with the center allele? Is it because they're linked in some way? And is it only linked genes that face this, that have this scenario?
Yeah, so the question is, how do we know that the flanking alleles are linked? The way I represent it is that they're linked. It's the same chromosome here, like this black bar represents the chromosome. And so you know that they're linked because you sequence the genomes nowadays. Like back in the days, there was actually a more difficult question. You would have to use proper genetic experiment, the stuff you did with Drosophila, to see whether the segregation is independent or whether it's not. And if it's not, you measure the number of centimorgans between the two loci here. But nowadays, it's just sequencing.
So you
You can only measure selective sweeps with link. No, you can do much more. This is a very shallow introduction to population genetics here. Like there are many other ways to measure selective sweeps, many other arguments. Most of them are based on intuitive ideas of what you expect from evolution. This is just one possible strategy, and that's really not the only one. You can make other cases, other arguments for selection. That's just the one that will matter for you in the context of this class. But yeah, there could be a million more arguments. It's up to your imagination, really. Other questions about this? All right. So let's continue with recombination because it's a difficult concept and it's one that's really important. You can think of recombination as a clock, as a way to measure time, really. So linkage equilibrium, is defined as the state where the frequency of the haplotypes are the product frequencies of the alleles. That means absolutely nothing. So let's try to see exactly what I meant here. Imagine you have two populations, a population where everybody looks like that with the pale alleles, okay, and another population where everybody looks slightly different with the dark alleles. And for some reason, there's a migration, like these two populations of equal size kind of meet and then they start to reproduce. It's the same species, they were separated for some reason, but now they'll live together and they start to reproduce. So what's going to happen to that population? Initially, you have only one of two options for the genotypes. Either you're pale or dark. These are the only two options that you have. But as time goes by, like the children of these individuals will start to mix, and then you will create new variants that did not exist before. Like the pale still exists, the dark still exists, but you also have now the dark pale and the pale dark. Okay, so you start to create with recombination new genotypes. So initially, just at the moment when the two populations merged, you had only two options, and you understand intuitively that with recombination and the children being born, then you will start to have new genotypes emerging. But they can happen only if recombination happens. Okay, so if these two alleles need to be broken by recombination, and that does not happen a lot, then you will have to wait several generations before these particularly new combinations, like dark, light, and light, dark appear, before you can see them present in the population. You will have to wait a long time that there are enough recombination events. Okay, and at some point, it will look at equilibrium. So, right before the populations merge, the frequency of the dark allele, this one, was 100%. Okay, just after the merge, it goes to 50%. You get that, right? Like in the beginning, in this population, everybody was like this. The migrants arrive. Now it's only 50% of the population that's dark orange. So the population is 50% dark orange. When you look at this over the time, the proportion of dark orange will remain 50%. This will never change. And the same for dark blue. It will remain 50%. What will change is the association of light orange and dark blue, and dark orange and light blue. Initially there was 0. Right after the merge there was still 0. It's only after recombination has happened that you start to observe more than 0 times these associations here. And if you give enough time, then the association of these new combinations will be the product of the frequency of the alleles. Okay? At equilibrium here, the light are 25%, because this allele here is at 50% and this allele is at 50%. So when you observe in the population that the combination of alleles is the product of their frequency, that's called equilibrium. That's the definition. Okay, and when you're at equilibrium, you stay at equilibrium. That's the stable state. Okay, everything else is called disequilibrium. So now that we can understand it, let's go. Linkage equilibrium is defined as the state where the frequency of the haplotypes are the product frequencies of the alleles. Okay, the haplotype is both alleles together, that's a haplotype, or multiple alleles together. If that here is the product of the frequencies, then that's equilibrium. 25% is 50% times 50%. Yes, you are at equilibrium here, there, and there, you're not at equilibrium. Okay? Everything else is called linkage disequilibrium in general populations, and linkage disequilibrium go toward equilibrium at a certain speed. So we can know when the disequilibrium started, the migration in this case. Okay, you get the idea now. Like in the beginning, things are 100% orange. At some point, it becomes only 50%, and they will remain at 50% forever. But what's going to change is the frequency of the cross alleles. Okay, and so if you find a certain frequency that is here, which is not at equilibrium, well, you can use that information to say I can compute when the disequilibrium initially happened, because that thing has a certain kinetic. Okay, you can see it as a timer. So this is one of the ways that we can measure migrations in human populations, because population coming from different areas of the world have got their favorite alleles that come with them, and when they start to reproduce, then of course that creates new combinations, and these new combinations, it's exactly that case here. They will initially create a disequilibrium. This disequilibrium will slowly erode, but until it has eroded, you can measure the time when populations have migrated. That's pretty cool, that we can use recombination as a way to measure time in terms of population genetics. So I'd like to show a couple of patterns now that are interesting for the genomes to know if they're selective or non-selective. One example of a selective pattern is the GC content in microorganisms. So the GC pairs are stronger than AT pairs. GC have got 3 hydrogen bonds, and AT have got only 2. Okay, so it's kind of harder to break a GC bond than an AT bond. You need more energy to break it. So this means that DNA with high GC content is less likely to be denatured at high temperature. The GC content of microorganisms correlates with the temperature of their niche. So here, that's on the x-axis, that's the temperature, optimal temperature of the growth of the microorganisms. And so remember that some of them are extremely happy in high temperature, like 80 degrees and more, like the thermophiles, and other are like temperate, 20 degrees good for them. And here you have the average GC percent of tRNAs. The tRNAs need to have a certain conformation, and the more the organism grows at crazy high temperature, the more G's and C's are in the tRNA. They need to have the proper conformation to remain functional. If they're open, it's not good for the tRNAs. They cannot work at all. And so there's a selective advantage in having many G's and C's in the tRNA so that they can remain properly formed at high temperature. You cannot remove too many G's and C's because you have to replace them by T's and A's. With only 2 bonds here, they'll break, and your tRNA is not going to be very useful. So the reason why there are more G's and C's when organisms grow at high temperature is understood, and that's selection. Those that did not died because the tRNAs were denatured. So that's an example of a genomic pattern that's due to selection, at least as far as we understand. And that's a very good explanation of what we see. The neutral pattern that is related is the fact that in mammals, like in most animals, but especially in mammals, CpG is scarce. So CpG means a cytosine, a phosphate, and a guanine. So it means the sequence 5' CG is just to differentiate it from G or C or C or G, etc. So people say CpG just to be specific. It means a C followed by a G. 5' is here, 3' is there. So methylated cytosine mutates 10,000 times faster than any other nucleotide. So here you've got cytosine, it looks like that. Methylated cytosine is in position 5 here of the cycle. You get 1 carbon and 3 hydrogens. So that's the methylation modification here of cytosine. So some of our cytosines are methylated in the genome, that happens. But when that happens, they will mutate to T 10,000 times faster than anything else. So it's like as soon as they get methylated, they get transformed into a T on evolutionary timescales. On the life of an organism, it's not the case, but over millions of years, if a C is methylated, it always turns into a T, always, and very fast on top of it. So it mutates 10,000 times faster than any other nucleotide. So in vertebrates, methylcytosine occurs only in the sequence CpG. So it's that cytosine here which is methylated. So this dinucleotide is very rare. The frequency is 1 in 100 instead of 1 in 16, because every CpG is transformed into TPG. Okay, the C mutates to T in that context. So concomitantly, promoters have no methylcytosine because it inhibits transcription. If you have methylcytosines, you can't transcribe. It's not a promoter anymore. So promoters can thus keep their CPGs because they're never methylated. They cannot. If they are, the organism dies. So it's selection in that case, which creates so-called CPG islands, where the dinucleotide has the expected frequency of 116. So if you look at the CPG nucleotides in the genome, in the human genome, they look like that. They're super rare in the genome and methylated. But in promoters, then they become not rare at all. They have their expected frequency of 1 in 16, and they're not methylated. Okay, and the reason why they're not rare here is because they're not methylated. Okay, like if they were methylated in promoters, they would instantly mutate to TGs with this explanation. Okay, it's because they're not methylated, then they remain CGs, and they're not methylated because of selection. If you methylate the promoter, the gene is not expressed and you die. So selection makes sure that they're not methylated, and then the fact that they're not methylated makes sure that they remain in the genome. Okay, that's a kind of a weird mechanism that is overall the fact that CPG mutates and is removed from the genome is neutral. The fact that it remains in the promoters is selection. So the same thing can be split into selection and a neutral evolution. We're going to make the break now, and we're going to come back to where we were before. So let's have a 5-minute break. We meet again at 3:05. I'll pause the recording for now. Okay, and we're back. So we continue with the neutral and selective patterns. And the next pattern that is neutral doesn't really have a name. I call it AA waves. So in eukaryotes, DNA is wrapped around nucleosomes, as you know. The dinucleotides APA, TPT, and TPA band easier, so DNA can bind tighter to nucleosomes if those nucleotides are 10 base pair apart. Okay, so DNA is kind of this double helix, and something that is 10 nucleotide apart will actually be exactly 1 turn over the DNA double helix. Okay, so if something is spaced exactly 10 nucleotides apart in a piece of DNA, it means that it will always be on the same position in this helix. And so imagine that if you have like AA nucleotides that are repeated every 10 base pair, then when you wrap this piece of DNA around the nucleosome, they're going to be on the outside, all of them, and that's going to make it easy to bend. So like the DNA can be closer to the nucleosome here, if you happen to have either AA, TT, or TA there. Okay, but there's no particular selection for that. You know, if fitness is not better if you have these AA nucleotides every 10 base pairs, but it will allow the DNA to be a little bit more tight, like snugly wrapped around the nucleosome. DNA that is tightly bound to nucleosomes mutates less because it is protected. So you have to see that when DNA is close to the nucleosome, it just mutates less. So if you happen to have AA every 10 base pairs, then your DNA is going to be more packed around the nucleosome, is going to stay like that. If you didn't have this AA 10 base pair apart, the DNA is going to mutate until the DNA can be kind of packed around the nucleosome. So that creates a bias of mutation, where you have an incentive for having AA nucleotides every 10 base pairs, just because once you get them, then the mutation rate is less. But that's still neutral, okay, even though that's biased, but it's still neutral because you do not have a selective fitness advantage in having these AA at 10 base pairs apart. You do not have more children because you've got that. It's just that your DNA is more wrapped around the nucleosomes and it mutates less. Okay, it's really important to understand the fundamental concept of neutral evolution. It does not mean that it's unbiased. It just means that you have no selective advantage in having the feature. It's a feature that can be biased. You know, it's completely possible, like these waves of AA every 10 base pair that we see in the genomes. But these, when you remove them, it doesn't matter. The organism doesn't grow slower, doesn't have fewer babies. Okay, that's not what it is. The pattern is coming from some other place than fitness. So DNA that is tightly bound to nucleosomes mutate less because it's protected. Sequences with APA, TPT, and TPA at 10 base pair intervals mutate less frequently. So this pattern is enriched in eukaryotic genomes. It looks like selection, but it is not. There's no fitness advantage. Okay, and you can see it when you align the pieces of DNA on the diode here, the place where the DNA enters the nucleosome, and you will see these waves. They're very clear in the genome of yeast, for instance. Now, something that is disputed, people don't agree whether it's selection or neutral evolution, is the GC skew. So what is the GC skew? It's defined as number of G's minus number of C's divided by the number of G's plus the number of C's on a given strand. Okay, so you follow one strand and you compute that score locally. In prokaryotes, the leading strand usually contains more G's and T's, whereas the lagging strand contains more C's and A's. Like many organisms have got that, but only bacteria and I suppose archaea, but I'm not sure. Okay, when you look at our genomes, it's not the case. It's only something that you find in microorganisms. So the neutral hypothesis is that the Okazaki fragments have mutational biases. Okay, like you follow one strand, and in bacteria you have one origin of replication, and so there's only one polymerase that goes from right to left, another one from left to right, and they meet somewhere in the middle. That means that there's only one leading strand for half of the chromosome from the origin till the terminus, and another leading strand on the other side. And the rest is like filled with Okazaki fragment, but the Okazaki fragments are always on the same strand on that half of the chromosome, and only on the other strand on that half of the chromosome. So the neutralist interpretation is that it's just a mutational bias. Like the Okazaki fragments are not repaired exactly the same way all the time. There are tiny biases accumulated over millions of generations of these microorganisms that creates this relatively random observation that this score is first biased positively on the first half of the chromosome and then negatively on the other half of the chromosome. So that's one interpretation. But people have never been able to show what the bias is experimentally when you measure the mutation rates in Okazaki fragments. So people who reject this hypothesis say, well, to be convincing, you would have to measure that bias and to show it to us that it exists. Right? Until you can't measure this bias, you can say it's whatever you like. The selective hypothesis is that many amino acids require a codon with a G or T in third position. Okay, you have to look at the genetic code, and you will see that for many codons, you need the G and the T, and so you need that particular amino acid in that position for your protein, otherwise your protein does not work, like a tryptophan or whatever that is. And so if you cannot get this G and this T, you cannot get your tryptophan and you're dead. So that's why you need these G's and T's on the genes. And because the genes are almost all oriented like the direction of the replication of DNA in bacteria, like that really goes like that, then they say, here you go, that's your explanation. It's just because you need these G's and these T's, otherwise you're dead. And so that's a way that you can explain why there are more G's and T's on the gene, is just because the genes are placed like that on the chromosome and they meet head to head or tail to tail at the end. Yes?
I was just wondering, is the G or T on the non-template strand? Because when it gets translated, you have to change that T to a U, I guess?
Yeah. So the question is, are G's and T's on the template strand or on the opposite strand? Just like now, I can't remember. I think the G's and T's are on the non-template strand. They're on the it means they're G's and U's in the RNA by memory, but double check. If this is important for you, double check. Oh, Yibing says that she's international. International. Okay, so that's an example of a pattern that is disputed. So people who reject the selective hypothesis say, oh well, yes, a second ago you told me to measure my mutation biases, but you should check your fitness. So like, show to me that when you remove these G's and T's, then your organism grows slower. And the guy said, yeah, but I have to change every G and T in the genome just to test that, right? I said, yeah, indeed, you have to. But you're asking me to measure impossibly small mutational biases of 10 to the minus 9 or something like this. So the discussion ends up like that. Like each of them goes into a place where the way to decide are just so subtle that it becomes very difficult to carry out. And then there's the mysterious isochores. People don't really know at all what's going on there. So many eukaryotes have large domains with high or low GC content. They're called isochores. The origin of isochores is still debated, but it seems that in vertebrates, they were created in the past, like a couple of million years ago, like 100 million years ago, and are now eroding. Prokaryotes have no isochores. In humans, isochores correspond to orangey bands, when you have the chromosome staining like that are visible on the karyotype. OR bands are GC rich, whereas G bands are AT rich. So the AT rich are the dark ones. Strikingly, most housekeeping genes reside in OR bands, while regulated genes are in G bands. So the human genome, and mainly mammalian genomes, at least in vertebrates, are organized in these blocks that are visible even when you look at the karyotypes. These are like, maybe you have wondered why do we see these bands like a zebra on the chromosomes? Well, the bands correspond to blocks of high GC or low GC. And within these blocks, you have, when it's high GC, you've got a lot of housekeeping genes. When it's low GC, you've got a lot of regulated genes. Why is it this way? Nobody really knows. It looks like selection, probably, I guess. But if selection, then what exactly is selected for? Why your fitness would go down if you don't have this organization? Could as well be neutral evolution. This may have something to do with the organization of enhancers, the fact that maybe genes share enhancers, so they need to be close when they're regulated, and there cannot be housekeeping genes in the middle, because then they would kind of mess up their own regulation. But that's purely theoretical. It's really unknown exactly what happens. So people are, of course, propose a hypothesis, or yell a hypothesis louder than other people. They say, I'm right, because I know it. But the truth is that we don't really know what creates the isochores, why they're this way. The only thing that seems to be clear is that they were created a long time ago, long time ago, and now they're disappearing. Like the GC content are slowly going back to equilibrium, slowly. But like something in our distant past, when we were not even a lizard, has created the genomes that look this way. Okay, so that's where we're supposed to have the break. Are there any questions on the differences between these features of the genome that are due to selection versus neutral evolution? But you get the main point, right? If it's neutral, it doesn't mean that this is symmetrical or unbiased. It just means that there's no fitness advantage in having it. It's something else that creates the pattern, but it cannot be a fitness advantage or disadvantage. Right? Is this idea a little bit clear? A little bit. All right. Any questions? Things you'd like me to go over once again?
Sir, I just kind of confused about the fact that it's not random for the neutrality. Could you expand on that a bit? Because when you talked about allele frequencies, you said if they evolve slower, then it's probably random. Does that mean that it's no longer neutrality, or?
Yeah, so the question is some confusion about neutrality and random, because when I was here explaining that if it's slow here, then it's random, then it's neutral. And so I've said it's got nothing to do with being random 2 seconds ago, so I'm self-contradicting. Yes, I am definitely self-contradicting a little bit. What I should have said is, if it's random, then it's neutral. Okay? If it's neutral, it doesn't have to be random. The criterion is not this one. Like something that is completely symmetric and random will obviously not cause fitness advantage, okay? Because it's going to be completely randomly or symmetrically distributed, so there cannot be anything to select for. But that's not the criterion. So if something is random, then it's hard to imagine that it would be associated to fitness. But the true criterion is whether something causes a fitness advantage or not. That will tell you whether it's selection or neutral. Okay? So yeah, that was a little bit confusing. Sorry about this. I think there was another question, right? Okay. All right, so we're going to go back to that slide again, because in reality, I know that most of you didn't get it. Understanding linkages equilibrium in just one slide is impossible. Even in one lesson is impossible. Like, that's one of the most difficult concepts to understand in biology in general, and definitely in evolution. And because you kind of need it for the class on GWAS, it's really important to understand a little bit how that concept works and why it's so important then I wanted to come back to it again randomly, just to ping your memory a little bit at the moment when you least expect it. So linkage disequilibrium, what is it really, truly? I mean, it just means that the genotype at one locus, that is to say a pair of alleles, is correlated with the genotype at another locus. That's all it is. Okay, if you have linkage disequilibrium, it means the information you have at that locus tells you something about the genotype at another locus. And it doesn't matter whether this locus is on the same chromosome or not. That's really all it means. If knowing your allele at one locus tells me something about anything else about your genome, then that's linkage disequilibrium. If knowing the allele you have at that locus gives you nothing else about your genome, then that's perfect equilibrium. Okay? That's really the concept of linkage disequilibrium. Do you learn something from one locus by knowing on another locus? So here's another example. That's another example based on the migration phenomenon, but I'm highlighting this time the fact that the population is diploid, and I'm grouping together the two alleles, and they're on different chromosomes here. Okay, so same scenario as before, migration. There were initially two individuals, very small population, gets to 4, and so these guys have random babies, etc. So what happens? At generation 0, people have, they've just met, they haven't had babies just yet. The new babies are created at generation 1. So look at what is possible. You can either have two people like this who had a baby, so the baby looks like that, two people like this who had a baby, the baby looks like that, or one cross, and the baby looks like that. So there are only 3 possibilities of babies, only 3. But in reality, with 2 alleles here on 2 sides, you have 9 possibilities, as you know. And yet, at the first generation, you observe only 3. Okay, you don't yet have the double dark, light dark, or the double light, and then light dark. These guys, you will have to wait yet another generation until these guys have babies together before they can appear. All right, you're still missing some of the genotypes. They haven't appeared just yet. Okay, so the answer is yes, you do have information about the genotype at another locus at generation 1, right? If you know the genotype is like this here, well, you know the genotype here at generation 1 is deterministic even. So you don't have a lot of information, so there is genetic linkage disequilibrium, sorry. Okay, so the loci do not even need to be linked on the same chromosome. However, linkage disequilibrium remains more than a few generations only if the loci are closely linked. So if they're on different chromosomes, like in this case, you do have linkage disequilibrium after 1 generation, but it's going to go to linkage extremely fast, exponentially fast. In just 3 generations, you're going to have almost no evidence of linkage disequilibrium. If the genes are together on the same chromosome and they're close, then it's going to last for generations and generations. Okay, so the concept is really, does knowing your genotype here tell you anything about the genotype somewhere else? If it does, there is disequilibrium. If it doesn't, you can say you're at equilibrium. All right. If you still don't get it, it's okay. It's a concept that's extremely hard to get around. So think about it, but that's really the idea. Does that give information on something else or not? So now let's see a little bit how we can build phylogenetic trees that I've been speaking about so far. So any phenotype can be used to build a phylogenetic tree. Like you have an example here with species A, B, C, D, E. And so you can group together the one that have whiskers, the one that have big ears, the one that have a fuzzy tail, etc. So all the ones in green have a fuzzy tail, and the A here doesn't have a fuzzy tail. So the ancestor look like that. So you can pretty much take any phenotype, anything you like, to make a phylogenetic tree. But which characters are most informative? Okay, like you took the whiskers, the ears, and the fuzzy tail, and you get a nice tree. But what about you had taken the paws, the claws, the color of the fur, something like that? Would you get the same tree? Maybe not. Okay. So sequencing gave rise to molecular phylogeny, where characters are sequences and phenotypes are the residues or nucleotides. So instead of using phenotypic characters like how big is the animal, what's the color, how many bones does it have in the hand, or something like that, you just use the sequence. You say, does it have a Q or a T? Does it have a Y or an A? And these are actually much more informative just because you get more variation. With just one gene that's represented here, doesn't matter what it is, but you can see that every nucleotide, or rather every amino acid or residue, is really giving you information about the evolution of the species. So you have as many characters to study as you have letters in the sequence, or these letters that are different. And so that's why it really brought phylogenetic to a new level when we were able to sequence proteins and to sequence genomes and DNA. Because before that, all the phylogenies are based on morphological characters, but at some point there just aren't any. You know, they're just not enough. You can say these two things, okay, they may be different at the species level, but they really look identical. Like they have exactly all the same phenotypes, right? So this is why nowadays very few people use the morphological characters, because you just have much more information at the sequence level. This is why these methods are so popular. So the way you do this is that you use MSA, or multiple sequence alignments. So there is no fast algorithm to solve the problem of multiple sequence alignment. It is much more difficult than the problem of pairwise alignment. So when you have two proteins to align, two sequence to align, it's a hard problem. You know, I spoke about the Smith-Waterman algorithm, etc. And when you have three proteins to align, it's not just 3 times harder. This is like much, much, much harder. And when you got 4, it's already a complete nightmare. So that's the problem. When you have more than 2 sequences, it's already a hard problem with 2 sequences, but it gets even much worse when you have more than 2. So therefore, one has to use heuristics that are not always accurate when the number of sequences is large. So it's still an area of investigation, how to make robust multiple alignments. At the moment, it's really impossible to explore all the possibilities to get the best alignment. The conserved residues or domains are usually the ones that have a function, but not always. So when you have an alignment like this, you can see in the beginning there's poor conservation, but some places like this nucleotide, or these residues are in orange, seems to be perfectly conserved between all the species. So you say, well, maybe that's important. Remember our criterion. If it's conserved, if biology decided that it cannot change it, maybe there's a reason. Like it's conserved because it's functional. That's always the same logic. So another important thing when you create a phylogenetic tree is to really distinguish the species tree from the gene tree. So when we decide we're not going to use the morphological features anymore, but we're going to use the sequence features, well, we say we're going to replace the evolution of the species by the evolution of their genes. So is that going to be the same thing? So a gene tree and a species tree should be the same, because the evolution of the species is the same as the evolution of the gene. Okay, you take one of my genes. So before the split between humans and chimpanzee, there was only one gene. Human and chimpanzee split. So there is my gene, there's the gene of the chimpanzees. And so I have evolved along that line. Chimpanzees have evolved along this line. My gene has evolved along this line. The chimpanzee gene has evolved along this line as well. So the species tree and the gene tree should be the same. Right? Upon speciation, 2 independent species evolve, and 2 independent copies of the genes evolve. The speciation has created 2 copies of the genes and 2 copies of the species. So that's why the phylogenetic tree should be the same. However, gene duplication creates 2 paralogs that follow separate evolutionary path. You can have duplication of the gene within a species. That's where the problem starts. Okay, like I'm me, I have suddenly one of my genes that duplicates, so there are now 2 copies of that gene, and I pass this on to my children. So even though they're still human, now the evolution of that gene has had an event, which is a split, the creation of a new gene that did not correspond to the evolution of the species. The species hasn't forked, but the gene has now a new copy. So after duplication, the species tree and the gene tree are no longer the same. So you have an example here. The duplication happens here in that particular species. So the light blue branches that are here correspond to the species, and the genes are indicated by these either red line or the blue line. So before the duplication, there's only one gene, but after the duplication, every individual has got 2 genes. But it's still one species here. Then you have a speciation event. Okay, like species goes this way or that way. It means now you have 4 copies of the genes. There are the 2 genes that went with that species and the 2 genes that went with that species. So the evolutionary tree of the species, which is indeed indicated with these like hazy blue lines, is no longer exactly the same as the evolutionary tree of the gene, which now looks like that. Okay, like you have the duplication here, and then the tree would look like that. So it is possible to date the gene duplication by comparing the gene tree and the species tree. If the two trees are exactly identical, there's nothing interesting. But if you have a difference, like the evolution of that gene here doesn't correspond to the evolution of the species, then you can be able to tell, well, obviously there was a duplication event of that gene in that particular species here, in that common ancestor between all these 6 species. And that is based just on the idea that after the duplication event, the two trees no longer have the same shape. So there are complications with paralogs. So genes can disappear as well. This can create inconsistencies between the gene trees and the species trees. So before they were different but consistent, and now they can become different and inconsistent. Horizontal gene transfer mediated by a virus or a transposon is another type of event that will modify the gene tree. It is possible to detect those events and reconstruct their history by comparing the gene tree and the species tree. Using the molecular clock hypothesis, we can even date those events, but it can be complicated. It should be can. So you have an example here, like you have the gene tree indicated here with copy 1, copy 2, copy 3. So there was an initial duplication here, and then one of the two genes has duplicated in one of the species, but then gene B disappeared, gene C disappeared, and gene A disappeared in different species. They just lost it. So at the end, all you have is something that looks like that, and the only topology that you can reconstruct would look like that in this particular branch here. Okay, so that corresponds to something that is incompatible with the evolution of the species. So you can guess, if you're good, that there was a duplication followed by a gene loss. But that's kind of difficult, and that's way above our pay grade for now. I'm just mentioning that so that you can know that there are methods when you study the evolution of species and the evolution of genes, but that you understand as well that since we're no longer very much following the evolution based on morphological criteria, you sometimes have these inconsistencies between the information you get from the sequence and the information you get from the logical evolution of the species. And you have to be wary of these cases. Another thing that makes it complicated to recreate the evolution of a species from the evolution of its genes is whole genome duplications. So there were 2 rounds of whole genome duplication in tetrapods and 3 rounds in fishes. So here you got the cephalochordates. They got 1 copy of the genome. All these animals, like mouse, the fish, and the lamprey here, they got 1 duplication here. Mammals got another duplication here. So we got 4 copies of every gene, being mammals, and fishes they got 8 because there was 1 more round of duplication happening early on in the fish lineage. Okay, so most genes have 4 copies, 4 paralogs in tetrapods, and 8 in fishes. So we have 4 copies of every gene, they have 8. Typically, every Drosophila gene has 4 homologs in humans, okay, because there was no whole genome duplication in insects, which means that when there is 1 gene in Drosophila, it's been duplicated 2 times on our lineage, so we got 4 copies of it. Many gene knockouts have no effect in humans because of redundancy among paralogs. Okay, whenever you remove one of the genes, usually you got 3 remaining that have relatively close functions, similar functions, so they can take over. This does not happen in Drosophila because they have 1 copy of every gene. When you make a knockout with CRISPR or with other methods, when they have a mutation, or double knockout, because of course they diploid, then the species, the individual, has much more problems than invertebrates and in mammals. Okay, so it's kind of weird to think about it, but at some point we have just doubled our genomes, and we got double the genes 2 times. In fishes, double the genes 3 times. So questions in the chat. But won't the new copies evolve to have new functions? Absolutely. They will. So in the beginning you get duplication, you get 2 genes, each of them will kind of evolve along its path. But because they had so much similarity to start with, like the structure of the protein will be very similar, then they will still overlap a little bit most of the time. And so that means that Whenever you remove one, the other one will be able to compensate for this for the phenotype. It won't be a pure compensation, but it will be something that allows you to survive most of the time. But it's hard to make to make very big blanket statements. Every case is unique, but that explains why knockouts are much more severe in insects than they are in vertebrates, for instance. Okay. So how do you build a phylogenetic tree? So like a gardener grows a tree, but in biology you build a tree. So building a tree requires a criterion. To say that this is the tree of evolution, you need a criterion, and the one that was traditionally used until recently was called parsimony. So choosing the tree that minimizes the number of changes is a possible approach. So you have 4 species here, and they can either be square or round. They can have 2 colors, like yellow or blue, and you have to make a scenario for their phylogeny. You could consider scenario 1, 2, and 3, and the parsimony criterion tells you that this is a much better tree. Why is that? Because you need to introduce only 2 changes. You need to say that here you went from square to circle, and here on that line you went from yellow to blue, and that's all you need. And you imagine that the ancestor here is looking like this, like a yellow square, and then you need only 2 changes. Every other phylogeny here requires that you have 3 changes or more. So that's the tree you choose. It's the one that says evolution changes, but it still hates changes. We're going to take the one that has the lowest amount of changes. We can reconstitute this as few changes as possible. That's parsimony, as few changes as possible. Nowadays, many trees are built with another criterion, which is called the maximum likelihood criterion. Almost the same, but it says that plausible changes are preferred over far-fetched ones. It's not just the number of changes. It says that some changes are they happen all the time, and some changes happen almost never. So you should give priority to the changes that happen all the time because they're not rare, but you should have a few rare events as possible. For example, replacing an amino acid that is a glutamic acid by an aspartic acid, that happens all the time. They're almost the same amino acid. So if all you have is like 7 of these changes in your phylogenies, yeah, it's fine, they happen all the time. But if you have 1 replacement by a tryptophan, then that's super rare. Like you never replace a aspartic acid by a tryptophan. So even though you would have 1 event, it'd still be less likely than 7 events that happen all the time. So that's the idea of maximum likelihood versus parsimony. That's of course a much better criterion because you make it more realistic. You say the changes are not all the same, some are likely, some are not likely, and I want to take the ones that are the most likely to happen. I don't want to take the rare events. So again, we have another problem when we want to build the trees, is that the number of trees grow very rapidly as the number of leaves increases. The leaves are the sequences of the species. Like either you're making a tree of sequences or a tree of species, and as soon as you got just a few leaves, being able to reconstruct the most likely tree or the most parsimonious tree becomes almost impossible. It is impossible to test all the topologies to find the one with the highest parsimony or likelihood. They are approximately 8 times 10 to the 12 trees with 15 leaves. Okay, here you got 1, 2, 3 leaves. Here you got 4 leaves. Here you got 4 leaves as well. So if I put 15 leaves, I would have to draw 8,000 billion of these trees. I wouldn't have the space to do it. I also wouldn't have the time. And even if I lived older than the universe, I still wouldn't have the time. This is really a big number. So with only 15 species or 15 sequences, 15 leaves in your tree, you really cannot reconstruct the best tree because you cannot investigate all these trees. There's just way too many of them. Okay, computers can hardly explore 10 to the 10 topology, and that's already very charitable. So tree reconstruction uses heuristics even for just a few leaves. So imagine now for the tree of life, when you have thousands of nodes on that tree, thousands of leaves, that becomes really hard to make sure that you got the actual tree of life. And we don't know what is the actual tree of life. We will never know. Really, there's no way that we can know that. Just way too many possibilities, and we cannot explore them all. We can get something that's close, but we'll never know whether we have exactly the right one. Another thing that you need to do when you build these trees is to root them, to place the roots, like the most recent, like the common ancestor. Phylogenetic trees are by default unrooted. The root is the most recent common ancestor, MRCA, that you'll see often. Its position on the tree does not change the topology, but it does change the history of the family. So you've got an unrooted tree here with A, B, C, D, and you got 5 places where the root can be, like this node in red. And if you put the root here, it means that this is the MRCA, the root there. Then that was the oldest version, was here, and then it gave a branch with AB on one side. So there was the first speciation event going left or going right. So going left, it created the species AB. Going right, it created the species CD, etc. So, but you could say, no, there's another possibility, this one here, and you could say that's the most recent common ancestor. It gives that phylogeny here. So the ancestor was actually quite close to C. The first speciation event gave one species here, C, and on the other side, it's got A, B, and D all together on that side. Okay, so you have as many possibilities of rooting the trees as you have branches. So when you have just that particular topology, you still don't know the scenario. You know relatively how the topology looks like, but you don't know what was the most common ancestor. The most common method to root a tree is to use an outgroup. That's a genospecies that we know is outside the genospecies of interest. So you have taxon A and B, like these 2 species. You just use something that you know is unrelated to them, going outside. That's why it's called an outgroup, because you know that it will have to branch on the most common ancestor of A or B. Okay, if you know it's outside, then that's a good outgroup. And when you have the topology of that tree here, you can remove taxon C and say, I just use it for knowing where the root is, but I don't care about taxon C. I know that the topology of my tree is rooted here. There are other methods as well that work when no outgroup is available, for example, for the tree of life. Imagine that, to know the origin of the tree of life, you cannot use an outgroup. You need something that's not alive. So you will never have an outgroup for that. So in that case, if you want to know where the root is for the tree of life, then you need to use a method that does not rely on outgroups. So with that, you can do very, very impressive things. The first you can do is ancestral reconstruction. When you know where the root is, where the most recent common ancestor is, you can try to reconstruct it, to know something about it. So when the root is set, it represents the most recent common ancestor, MRCA, of the family. One can use ancestral reconstruction techniques to guess the sequence of the MRCA. On the left is a tree of bacterial elongation factors. The authors of that study synthesized the MRCA in vitro and measured their melting temperature. Okay, so once they got the tree, once they've got the root, they can say, oh, well, we know this, we can guess what the sequence was of these species that were here. They disappeared billions of years ago, they don't exist anymore, but we can still guess what they had because that's consistent with the topology. Okay, like it's a bit playing Sherlock Holmes. And so they have taken these sequences where the arrows are, and they have synthesized them in vitro. Like, we can do that now, it's not a problem at all. Like you create a synthetic pieces of DNA, put it in a bacterium, and then it produces the protein. And they measured the protein, the stability of the protein versus temperature. And so what they show here is the temperature on the y-axis of these proteins that they have synthesized in vitro. And what they show here on the x-axis is the time of the most recent common ancestor. You can see that it goes to 4 billion years. I remind you that life is approximately 4 billion years. Right? So that goes to the origins of life, super long time ago. And the temperature that they show here is the one that they measured experimentally with this method here, but that's also the temperature of the oceans that is here shown like that. So yeah, 4 billion years ago, the oceans were approximately 80 degrees. That's pretty hot. So that created a very different set of conditions for life to emerge. It was not really the same as now, very far from that. And you can see that there's an amazing match between the temperature of these proteins that they have reconstructed and what we believed was the temperature of the ocean at the time when these proteins existed. They no longer exist, they're all gone, all of them. Okay? So on the left is a tree of a bacterial elongation factor. They also synthesize the MRC in vitro and measure the melting temperature. Next to it is the correspondence between the inferred temperature of the oceans, that's the dotted line, and the stability of the protein. Another case where people have used this ancestral reconstruction is to recreate a transposon that was long dead. The transposase Sleeping Beauty belonged to a dead transposon found in fish genomes. So they used the same method, made the evolutionary tree of all these transposons that they found in fish. The transposon was dead in the sense that all of them had stopped codons everywhere. They were not really functional anymore. They were not making any copies of themselves. But by making the tree, they could say, well, the ancestor must have looked like that. So they recreated the ancestor. So now they had a transposon that was functional, even though it was physically dead in modern days. And the transposon started to jump again. And so they called it Sleeping Beauty, of course, because it was not really dead. It was just sleeping for a couple million years. And so until 1997, it was dead, and then it was resuscitated in 1997. And this is still by far the most active transposon that is known. It's very useful for biotechnologies because this enzyme is the most active transposase that we know today. So that's showing just one example of biotechnology using evolution to create some tools that are useful today. You can also reverse the trees not just in time but in space, because things diffuse, of course, over time, but species and everything that's alive really has a place in space as well. And you could say, if I reverse time to know when was the most recent common ancestor, I can maybe ask where was the most recent common ancestor. So duplication and speciation is accompanied by migration, of course. So one can combine the ancestral reconstruction techniques with geographic data to map the origin of a phenomenon. On the left is the proposed evolution of Indo-European languages from their putative origin in Anatolia. So you can do this with genomes, you can do this with morphological features, but you can do this with languages the same. So you take words as proxies for genes, and you just plug this in the method. This is actually what the guys did. They used software that was for genomics. And you have the languages of Europe and India here with the Celtic, Italic, etc. And based on the information that we have with the common version of languages, you can infer the most recent common ancestor being this one. Okay, so here's the root. So that's the primitive Indo-European language. It's the one that has this group of brown languages here, Anatolian on this side. And because this must be the common root, and because of the distribution of people now, they say that the people who originally spoke Indo-European must have lived here in Anatolia. So that is now the most commonly accepted answer, but it's debated still. When people don't agree, they say no, that method is not the right one, we should use another one. But slowly there's more consensus for that being the place where people who speak either the European languages or the Indian ones, not all the Indian ones, are from here. Another way this can be used is to date extremely old events like Adam and Eve using structured coalescent techniques. That's pretty much the same as what I was showing before, reconstructing the ancestor, but not only in time, also in space. It is possible to find the most common ancestor of all Y chromosomes. Okay, like you take the sequence of Y chromosomes today, you just go back in time to say what was the Y chromosome looking like in the beginning, and actually where was it? You just look at where are the people today, what kind of SNPs or variations we find at the different locations. And so they say, well, the father of all Y chromosome was the oldest man. That's the father of everybody here, because to some extent we are linked in a very real way, that my great-great-great-great-great-grandparents are your great-great-great-grandparents. Okay? And everybody at some point is from Africa. And so this is what they call Adam, the father of all Y chromosomes. They did the same with mitochondrial DNA, and because DNA from the mitochondria is passed by mothers, then they found Eve, and both of them are in Africa from more or less the same place. They were very happy about this, except that Adam never met Eve because they were living 100,000 years of difference or something like that. So, this biblical view, of course, has got nothing to do with science. So that has, they call this Adam and Eve, I hope, for fun, because this is absolutely not based on any reality given the Bible. It's just that, yes, there has to be a most common ancestor of all Y chromosome. Yes, that person who had that chromosome is literally the father of every human at the moment, definitely. But at the time he lived, other people lived as well. So it's got nothing to do with just Adam and just Eve in the middle of Africa. It's just that people who were living at the same time as Adam just were not lucky to reproduce as much as he did. And the same for Eve. And Eve doesn't have to be next to Adam in the middle of Eden Garden. Okay, so it's just like genetic views say, yeah, there is a common ancestor of the Y chromosome and of mitochondrial DNA, but it doesn't have to be that they were alone at the time. It doesn't have to be that they were together. Okay, I hope that's clear. Another case that has been cracked with this method, which is quite interesting, is the emergence of HIV. So HIV emerged in Kinshasa, in the Democratic Republic of Congo, around 1925. It's now known. It reached Brazzaville in the Republic of Congo just a few years later. It followed the rivers toward the east and the train track toward the west. Okay, so this is where HIV, the spillover of HIV-1 happened. And now, just based on these methods of going back to the ancestor in time, you can know when that happened. And that happened in 1925, give or take a couple of years. So that's pretty accurate. And also doing the same in space, because where did that happen? That happened here in Kinshasa. And then you can play the movie forward a little bit to see that very rapidly HIV spread along the axis of communications because people were traveling, of course, and this is how HIV spread in the beginning. So here there was a spillover, probably from hunting monkeys. This is a very bloody activity when you had monkeys. This is why people imagine that, yeah, you can easily get infected by monkey blood because this is a very violent act here over the chimpanzees. But it's clear that this has been an event that happened a bit longer than we thought, because in the beginning we started to realize that there was a new disease only in 1981. Let's say 79, 80 were really the first years, but the virus had been already going around for quite some years. So this is how this can be very useful for understanding diseases, to do this kind of analyses. Of course, the same happens with SARS-CoV-2, as you probably imagine, and part of the data is not available, but still things are pretty consistent with the Wuhan being the source of the epidemic. So there's also non-phylogenetic trees. It's important to understand that just because it's a tree does not mean that there's a phylogenetic relationship between the branches. So trees are also used for clustering. Such trees, called dendrograms, so it's not a phylogenetic tree, it's a dendrogram, are not phylogenetic. They only indicate possible groupings between the objects based on their similarity. Left is a dendrogram of US states compared by their arrest profiles, number of murders, assaults, rape, and total urban population. Okay, so people measure a couple of crime indicators like number of murders, etc., and they put the 50 states, 51 states in some sort of grouping, and they want to say, okay, there's the states that look like that, the states that look like that, so that we can have a profile of criminality in the state. Is it white crime color, not white crime color, etc. And so then, once you have groups, you can also make groups of groups, and you can make groups of groups of groups, and that gives you a tree in the end. But it's not an evolutionary tree. It's not like Alabama had a common ancestor with Minnesota that's split. It's not at all what happened. Okay. So just because it's a tree doesn't mean that this is phylogenetic. There are other reasons to make trees based on similarities, but they're not always phylogenetic. So the classification in 4 groups is highlighted. Classifications with more or fewer groups are also possible. So it's just for clustering. Don't get confused between a dendrogram and a phylogenetic tree. So that's what I wanted to tell you today. The key points to remember is that conservation highlights physiological relevance. Okay, it's pretty clear. Nobody will say it in those terms. It's probably a little bit too simple, but really, if it's conserved, it's important. It's relevant. If it's not conserved, people will tend to think it's not. Okay, it's not always the case. Bear in mind that some things are meant to evolve fast. That's the way they work. But in general, the overall belief is, if it's conserved, it matters. It's worth studying. Phylogenetic trees allow us to infer the past. And with these methods of ancestral reconstruction, we can look into the past to see what has happened, how did these things evolve, how did they occur. That's really a tool for archaeologists. We're really interested in the past when we have a tree. We're not really interested in the future that much. And phylogenetic methods are algorithmically very complex. They're much more complex than everything we've seen so far. So it really takes dedicated studies and machinery to even understand how to reconstruct trees or how to make multiple sequence alignments. That's it for today. So are there any questions with what we've seen so far? I'm sure there are. It's one of the hardest class, this one. Like the concepts where I'm from, these things are taught in second year. But apparently here you have zero exposure to trees and evolution in these quantitative terms. So I was told last year that most people were not familiar with the concepts I presented. I think it must be the same this year. So it's okay if you're confused. Completely okay. Just take the time to go through it again, and you'll see there's nothing really complicated about it. But the first time, it's a lot to take. Well, if no question, I have there are questions. What exactly does common ancestor of all Y chromosomes mean? Is it the first organism that can undergo sexual reproduction? No, not at all. Not at all. Very far from that. So you take all the Y chromosomes in the present population. Like we all men inherit the Y chromosome from the fathers, which also inherited from their fathers, and so on. So at some point, there's a guy, a real guy. I mean, like it's not an imaginary person. Like it was a real guy, who is the father of all human beings. Believe it or not. So like your great granddad is my great granddad. It has to be the case. Like there's no other possibility. So that's what we mean by the common ancestor of all Y chromosomes. It means that the Y chromosome of that lucky dude, okay, has passed to literally every human now. I mean, every male, because women don't have Y chromosomes, of course. Okay, so that's why it's the most recent common ancestor. It just means that when you trace back the lineages of all the Y chromosomes of human in the present population, at some point they go back to a single guy. And so that's the definition here. So functionally, that's way after reproduction. Okay, like for example, this person lived after the split between monkeys and chimpanzees and humans. So like it was really a guy. It happened like approximately, if I remember, 200 million years ago. So he was looking already like modern humans mostly. Does it account for interbreeding? It accounts for everything. Okay, like you just take human population now. There is a guy that is the father of everyone. I mean, the father, like the grandfather of everybody. It has to be the case. And the same on the mother's side. There is a woman who is the lucky grandmother of the whole planet. And that's the definition. It's an operative one. It's not a functional one. It's just that it's a necessity. It has to be the case that at some point when you go back in time, we all are from the same father and all are from the same mother.
Sort of a random question. Considering that they are all algorithmically complex, sorry, my brain is slow today. What do you think about those 23andMe, those ancestry tests? How accurate can they be when you get them in such a quick amount of time? Or if you're coming from a place where maybe documentation is not as, like common census and all of that, what do you think about the accuracy of those tests?
So the question is what I think about the accuracy of genetic tests like 23andMe. I don't have a strong opinion. I never did it, so I don't know exactly how reliable that is. I don't think that there is a huge issue with the getting the right SNPs. I think that by now the methods are relatively straightforward and they're relatively cheap. But they tell you, oh, you're 58% from Germany and 38% from Italy. Okay, what does it mean exactly? There's a huge amount of interpretation of what that could possibly mean, and I haven't seen any explanation anywhere. People seem to be happy to just know that they're 58% German. But you say what could that possibly mean? Does it mean that the SNPs that they found were SNPs that were born in Germany? That were like when you do the reconstruction, they say this SNP must have happened for the first time in Germany. Or does it mean that this is a SNP that is often found in Germany today and no other populations? I'm not sure exactly what that means. So I don't really have an issue with the precision. Maybe Michelle will tell you more about this next week because she's more of a human geneticist than me. But I would like to see more information about what they call being from a certain place or the X% from somewhere else or something like that. We have 1 more minute for burning questions, if any. Well, if no further question, then I'll just quickly go to the exercise that we could have done. But no, there are questions actually. So, how do we account for interspecies breeding in phylogeny? It's a good question. The answer is mostly we don't. We hope it doesn't happen because that becomes difficult. The same as horizontal gene transfer in gene phylogenies. You know, that really creates a mess. And now we have to leave the room, so I'll try to answer the last question very fast. Understand that the Y and X chromosome phylogenetic tree leads to Eve and Adam. My question is, are we skeptical that they lived together because there was 100,000 years apart? If yes, did he come before her? Is it possible that he was that much older than him? I'm not sure how to answer that question. Now we have to leave the room, so if that's okay with you, Rashid, I'll answer that question in private. So thank you very much, everybody, and see you next time.